And now it's a great pleasure for me to introduce our second speaker today. Christien Betz is the head of molecular genetics at Bioscientia. Bioscientia is, I guess, the biggest diagnostic lab in Germany. And he did his PhD in molecular biology at the Ludwig Maximilian University in Munich. And since 2014, he is responsible, as I mentioned, for molecular genetics at Bioscientia. He is also responsible for the research and development team, and especially for the next generation sequencing group. And Christien is a real expert, especially also in establishing new technologies in, I would say, clinical routine settings. And I'm really happy that he will tell us today a little bit how Bioscientia established our whole exome workflow for the analysis of genetic diseases. Christien.
Thank you for the nice introduction. As Bernd has already mentioned, we are at Bioscientia a network of laboratories that are placed over all of Germany, more or less, but we are part of an even bigger network of laboratories. We are part of Sonic Healthcare, and in this network we are one of the biggest human genetics laboratories or departments, and in this laboratory we're doing a whole range of different analyses, of course. For example, we have a booth here at the ESHG that is focusing mainly on long-read whole genome sequencing, our newest application, but the backbone in our molecular genetics department is still whole exome sequencing, and I will explain today how we have established it and at what. It's a really application-based approach to you, how our lab is run, how we do the best throughput and the cost-effective use of an exome-based approach. So this slide shows more or less the evolution of resolution, showing both more or less our portfolio in the lab from cytogenetics, which also pronounced dead many times, is still half of our department, and we don't see any shift in a reduction of samples. Of course, here you look at a chromosome level of analysis, while in the molecular genetics department we are mostly doing sequencing, and here we have really the whole range of different NGS approaches, starting with panel sequencing, which we did before we shifted to exome, and now we use exome as a backbone for all our NGS analysis, and combine that with long-read whole genome sequencing for future applications. We are a long-term Roche customer in regards of the consumables for our exome analysis. We use both the chemistries from Roche for our library preparation as well as for the enrichment process of the exome. Before we use the current HyperExome product, we were customers of the MedExome product, and already now 4 years ago the switch happened to the HyperExome. Since then we have processed more than 17,000 samples with the version 1 of this product, and for about a year now we have implemented the update to the version 2, and since then I've processed about 6,500 samples, so our monthly throughput is between 500 and 600 samples approximately. In combination, more than 23,000 samples have been processed, analyzed, and also fill our internal databases, of course, with all the variants we detect. So why are we still doing exome sequencing when we can also do whole genome sequencing, where we can look at many more things, of course? As a commercial lab, we have to look also at turnaround times and also at cost, of course, and while the human genome has more than 3 gigabases in size, the exome, meaning the combination of all coding exons in all known genes combined, are just about 43 megabases. This is the size of the capture oligos in total that is used by the HyperExome, for example. And you see by the difference that you need to sequence much less to get information from those regions. We actually have 2 NovaSeq 6000 instruments in our lab using the S1 flow cell, and with this flow cell you can either combine 4 whole genome samples or 40 exome samples, and in our work through trying to find a compromise between turnaround time and cost, we usually load 40 exome samples on these flow cells and have like this a really cost-effective way of producing. When so many target regions are combined, of course, it is important to have some kind of balancing of those capture oligos. While the approach of whole exome sequencing is definitely to cover all known exons, the main emphasis is also to cover, of course, regions that are known to play a role in human disease. Therefore, the known databases containing such variants were all used by Roche to actually be sure that the content there will be part of the enrichment oligos and therefore present in the sequence data that you produce, especially HDMD, for example, RefSec, but also ClinVar. We all look to when we check our variants for pathogenicity. And in those databases, of course, there are not only coding variants, but also in regions that are intronic, for example, and even those contents were actually used by Roche to be added to the content. So even if a known pathogenic variant would be intronic, you could still cover it with the exome analysis. Of course, you will find a lot of disclaimers on the slides that the product is for research use only. We have used this product in our lab to use it as a lab-developed test, did all the validation here, so we were able to use it in diagnostic purposes. As I've mentioned before, so many different target capture oligos combined have always the problem that you need to do some kind of balancing. You need to make sure that each exon is represented and as even as possible, since you don't want to do only SNV analysis with this, but also CNV analysis. And problematic is, for example, if an exon has a low coverage while all the others are there that you're interested in, or like in point C, you have a missing exon, especially if there are known pathogenic variants in such a region, it's not part of your analysis. You will always have a problem of being blind there and have to do extra measures like Sanger sequencing of that single exon to also be able to cover that. To check, to compensate for a low covered exon, of course, you can increase your overall coverage. Of course, then you also have a cost problem because you over cover the exons that are already nicely covered, while the missing exon is, of course, not improved at all by that approach. Therefore, luckily with the HyperExome, we have a product that where this balancing was done with really good, or actually great performance. So we are really happy with how the exonic sequences are covered in this product. Typical workflow is, of course, for this NGS approach to start with the extracted DNA. In our case, most DNA is from blood, but it can be other tissues as well. You do your library preparation, after that library preparation, the target enrichment. This is where the HyperExome comes in place. After QC, you do your sequencing. Our typical workflow is actually automated. We use 2 Hamilton NGS star instruments with batch sizes of 40 to 48 samples. Our multiplexing in the hybridization is done at 20 samples. And these 20 sample pools can then be either loaded separately on each lane with the XP workflow on one S1 flow cell, or you can do a combination of 40 samples and load them in the standard workflow together. And then the standard short read sequencing would be 2 times 150 base pair sequencing paired end. The 2 instruments next to each other have the advantage that if you have a downtime of one instrument, you still have the other one available. And the throughput is, for our purpose, well enough. You can do several batches per day if needed. And the bottleneck is definitely not the library preparation. Typical hybridization is still done overnight in our laboratory, although there are protocols available that have similar results even with just a few hours. But this is, again, not really a constraint. And library QC quantification is done with that pool afterwards before sequencing. And usually we get really nice insert sizes where we want it. And good coverages. Usually we aim for 100x and get that nicely from that S1 flow cell with 40 samples. The hybridization with this HyperExome works that these complementary target oligos called Betz or capture oligos are consisting of 120 base pairs, and they are designed, of course, to these regions of interest, usually the exonic sequences, including the exon-intron boundaries. So the 10 bases plus and minus are usually covered nicely. These baits are labeled with biotin, so you can actually fish for them with streptavidin labeled magnetic beads. And they are then, after magnetic binding, several stringency washes are performed to wash away those DNAs that have not hybridized to these capture oligos. And then with a low salt, usually water or low TE buffer, you can release those captured DNA fragments, and this is then your ready library. With this approach, a on-target rate to the exonic sequences greater 80% is very typical, which is also nicely for cost-saving sequencing, because you don't get a lot of off-target sequences. Our newest addition in the lab is the Roche AVENIO Edge instrument, an instrument you can also look at the Roche booth currently, until tonight at least. And we have that for evaluation in our lab currently, and this is really a nice improvement even over the Hamilton robots. It can do the whole setup if wanted. We can start with the DNA samples. It can do the complete library prep. It can do the pooling. It can do the quantification. And it can do all the hybridization and washing if we want that in one go. And especially if you want to use like the weekend to create your libraries. This is a really off-hands approach of being able to do that. In our hands, one instrument would not cover all the throughput, but we use it like currently for at least 25 to 30% of our samples. And from the feedback I get from our technicians is that it's almost too simple to operate. It uses cartridges for almost everything. They are color-coded with the reagents and also bar-coded, so the instrument can check actually its correct loading and positioning. And once this is done, a test run with the scanner actually makes sure that everything is loaded in the way the instrument expects it to be loaded. To avoid errors later in the run, you get nice signal lights, actually like a green, meaning everything passes and can start. And for the future, actually, I think we would definitely like to replace our Hamilton robots with such a more complete system that can do even the quantification on deck. Currently, with the Hamilton robots, we do that off deck. And since we do 20 plexes for hybridization, we do that also manually. That exceeds actually the recommended amount that Roche recommends by a few samples, but it's really working in our hands greatly. Sequencing, as mentioned, is done on our two NovaSeq instruments, which we frequently get more than 700 gigabases actually output from one S1 flow cell with this approach, meaning the pools of 20 samples or 40 samples, depending if XP workflow or standard workflow, all are at least 100x covered, some even up to 150x. We have implemented, of course, a lot of quality control steps. We have the advantage of our own bioinformatics team that has developed tools for us that help us in an automated workflow, that also automated quality control. Things we look at is, of course, mean coverage, but also how does the pool look, how even are the samples distributed in the pool, how much is actually at least 15x covered of all regions, and what is the on-target rate. In this case, everything is above 75%. And other quality control steps are, of course, like the gender that is checked, is it the correct sample, is the index adapter the one that is expected, and we do a contamination check. I think in the next slide I will show that in detail. For example, with the zygosity of variants you would expect homozygous and heterozygous variants, of course a few mosaicisms as well, but the majority of variants would be at 50% and 100%. And in case of a contaminated sample, you would get additional peaks in the 75 and 25% region. If this is present, our QC caller would actually call this sample failed, and we would have to look where this contamination happened. Another thing is, of course, sample identity check. For each sample we run with the HyperExome, we do a second DNA preparation that is run in a different workflow with 24 SNPs that are checked with a CASP as a competitive allele-specific PCR that is run by qPCR, and this SNP results are then compared with the NGS data, and should there be at least 20 out of the 24 SNPs have to be identical. It is allowed for a few to drop out, but they should never show opposite results. This is always a hint that there might be a sample mix-up at some point, and the algorithm our bioinformatic has designed actually compares that SNP result with all other samples we have sequenced so far, and would actually find a close match for us to also help us identify should there be a mix-up, where did that happen, and what is the probable cause for it. We are so happy with the HyperExome that we have switched our NGS workflow completely away from panels and use the exome as a backbone for virtual panels. We filter just bioinformatically from the exome data. Of course, the best analysis is done if we also have the parents available for our index patients, so we can also call variants as de novo. And the value by doing a streamlined approach in our lab is so good that we can even use it for just a single gene analysis or even a few variants if we compare the time involved to actually design primers for a Sanger confirmation of certain familial variants where we know where to find them. For a single variant, we would not run exome, but as soon as we have to check for a handful of variants, this is already an approach which we think is at least time-saving and not much more expensive. The distribution of coverage is so even that our CNV analysis is actually also quite nicely done. I show some examples later, but even for single exons, we have detected a few clear, true deletions that the coverage data already hinted at. And we offer really a huge range of panel diagnostics, which you can all find on our website. All of those are actually generated from the exome as the backbone. Our bioinformatics team has, of course, implemented some nice tools for us, so we can analyze the data from the exome nicely. We have our own internal frequency database. We have this QC database. We have a panel database where we can update the content on a daily basis if needed. We have a phenotype database that helps us for our automated report writing, meaning we can select a variant that we would like to have in the report, and the phenotype for this disease in the database would already generate a pre-written report that we just have to adapt to the individual. A primer database helps us for confirmation by Sanger sequencing if needed. And yeah, I think, yeah, this is more or less a nice overview. When switching from medexome to the HyperExome, we had really a good improvement in the quality of our exome analysis. However, version 1 was not perfect. We have detected several exons also in known disease genes that were not covered there, but we had extensive talks to Roche about that, and almost all, I think, the input we gave there was actually implemented in the version 2, and now all those regions that we were aware of are now nicely covered in version 2, and we have no really reason in complaining currently. One thing we always like to analyze in addition to the genomic exons are, of course, the mitochondrial sequences, and that is not covered in the exome sequencing. Therefore, we previously did a custom design that we added to the whole exome to be able to cover that as well. However, we found a nice workaround in the laboratory where by adding a small amount of non-enriched library to the enriched pool of libraries after the hybridization, we expand the covered regions a little bit, but enough to cover the mitochondrial genome completely in at least 30 to 50x. This is a very cost-effective way to also get sequences for the mitochondrial genes. And we have implemented that in our workflow, and since then do not have to do special extra analysis to cover the mitochondrial genome. Sanger sequencing of missing exons we don't have to do anymore since version 2 is published or available. Here, now some examples of now covered exons that were previously missed. One example is here in CNGB1 exon 14. Oops. Not sure if this works, but you just see it there. That in the red marked area, you see version 1 on the upper half, version 2 in the lower half, and you just see it is now covered where there was just a gap before. A similar thing in NF1, exons 20 and 23, now present in version 1, a gap. And this is actually an example of an intronic variant where you see this. The upper blue mark means that it is in the HGMD database, in this case also in the ClinVar database, a pathogenic intronic variant, not really part of the whole exome, but it has a bait designed to it, and it can be analyzed with the exome analysis, although intronic. In this case, this is a known disease gene for Stargardt disease, ABCA4. So those regions would also be available with the HyperExome. And as mentioned before, the even distribution of coverage allows for a nice CMV detection. In this case, this was a single exon, heterozygously deleted, only one allele left. By just looking with your eye, you hardly see a difference in coverage in the upper individual compared to two randomly selected normal individuals below. However, we were able to confirm this deletion that our caller actually clearly called by MLPA. So even a single exon can be detected in a CMV analysis. However, we would always do MLPA confirmation if available for single exons. For 3 consecutive exons, we feel confident enough to be able to call them without further confirmation. So where are we currently with doing this kind of exome analysis? We are already there that exome analysis replaced previous panel or single gene analysis. We do one stream, one workflow, really streamed workflow in the lab for all these kind of analysis. Our tree analysis really has a high diagnostic yield. However, since we have tasted the quality of long read whole genome sequencing, which is of course still 4 to 5 times more expensive than an exome analysis, but gives you access to regions that are previously not covered at all, with decreasing the sequencing cost in the future. It could very well be the future that we all do long read whole genome sequencing in the future, replacing the current exome sequencing. However, currently still, version 2 of the HyperExome is our standard analysis, where I would say more than 80% of our samples are run in the molecular genetics department. With this, I would like to close for today, and I think we're open for questions if any.